Welcome to this lecture on Genome-Wide Association Studies, often abbreviated as GWAS. Today, we are going to explore one of the most powerful tools in modern computational biology. We will learn how scientists sift through billions of letters of DNA to find the exact genetic typos responsible for complex diseases. By the end of this video, you will understand both the biological intuition and the mathematical foundations that make these discoveries possible. Now that we know what we are setting out to learn, let us start with a simple analogy. Imagine you have two massive libraries, each containing thousands of instruction manuals for building a human being. One library belongs to people who love eating cilantro, and the other belongs to people who think cilantro tastes like soap. If you wanted to find out why they differ, you would not read every single word of every single book. Instead, you would quickly scan specific pages where you know typos usually happen. If you notice that the soap group almost always has a T printed on page 45, while the love group has a C, you have just found a genetic link to that trait. This is exactly what a genome-wide association study does on a microscopic level. Building on our library analogy, let us look at the formal biological definition. Formally, a genome-wide association study is an observational study of genetic variants across different individuals. The human genome contains billions of DNA letters, but as humans, we are about 99.9% identical to each other. The tiny 0.1% differences are mostly single letter changes in our DNA code, called single nucleotide polymorphisms, pronounced as SNPs. In a GWAS, researchers use advanced technology to scan millions of these SNPs across thousands of people's genomes simultaneously. They use statistical tests to determine if a specific SNP occurs significantly more often in people with a particular condition, like diabetes, compared to people without it. If a strong link is found, that SNP is associated with the disease. Understanding what a SNP is naturally leads us to ask, why is scanning for them so incredibly important? Before GWAS was invented, finding the genetic cause of a complex disease was like searching for a needle in a dark room without a flashlight. Diseases like Alzheimer's, schizophrenia, and type 2 diabetes are not caused by a single broken gene, but by hundreds of tiny genetic tweaks working together across the entire genome. GWAS acts as a massive floodlight, allowing scientists to scan the entire genome at once and pinpoint exactly which regions are contributing to the disease. This has revolutionized personalized medicine, allowing doctors to predict a patient's risk based on their unique genetic profile, and helping pharmaceutical companies develop highly targeted drugs to treat the root cause. Now that we understand the immense value of these studies, let us look at how they work mathematically. To figure out if a SNP is actually linked to a disease, we use a metric called the odds ratio. Conceptually, this formula compares the odds of having the disease if you carry the risky genetic variant to the odds of having it if you do not. Let us break down the symbols. OR stands for the odds ratio, which is a unitless number. The variable A represents the number of sick people who have the risky variant. B represents healthy people who still have the risky variant. C represents sick people without the variant, and D represents healthy people without the variant. If the resulting OR is exactly 1, the variant has no effect. If it is greater than 1, the risk increases, and if it is less than 1, the variant is protective. Before we move on, let us see this formula in action with a concrete example. Let us say we are studying type 2 diabetes, and suspect the A variant increases risk. We are given data for 1,000 diabetics and 1,000 healthy people. Among the diabetics, 600 have the A variant, which is our A, and 400 have the G variant, which is our C. Among the healthy controls, 300 have the A variant, our B, and 700 have the G variant, our D. We chose the odds ratio formula to directly compare these cross products. Substituting our numbers, the numerator becomes 600 multiplied by 700, giving 420,000. The denominator becomes 300 multiplied by 400, giving 120,000. Dividing these gives a final answer of 3.5. Physically, this means people carrying the A variant have 3.5 times higher odds of having type 2 diabetes. This is a strong genetic association. This impressive odds ratio of 3.5 leads us to an important scientific question. Even though the number is high, could this result have happened just by random chance in our specific sample of 2,000 people? In science, we cannot just trust a ratio. We must prove its statistical significance. To answer this, genome-wide association studies use the chi-square test. This test evaluates whether the difference between the numbers we actually observed and the numbers we would expect to see by pure chance is statistically significant. By applying this test, we ensure that the genetic associations we find are robust, reliable, and not just a random fluke in the data. Now that we know why we need to test for significance, let us look at the mathematical tool we use. This is the chi-square formula, specifically a shortcut designed for a 2x2 table. Conceptually, it measures how far our observed data deviates from the expected baseline. Let us define the symbols. The Greek letter chi squared represents our test statistic. The variable n is the total sample size of our study. The terms A, B, C, and D are the exact same group counts we used in the odds ratio. The numerator squares the difference of our cross products, amplifying any real genetic effect. The denominator is the product of all the marginal totals, which normalizes the result. A larger chi-square value means the result is less likely to be due to random chance. Having seen the formula, let us apply it to our diabetes example to see if our odds ratio holds up. We are given the same data, so our total sample size n is 2,000. First, we need the marginal totals for the denominator. The total number of people with the A variant, A plus B, is 900. The total with the G variant, C plus D, is 1,100. The total diabetics, A plus C, is 1,000, and total healthy people, B plus D, is also 1,000. We chose this shortcut formula because it saves us from calculating expected values individually. We substitute n and our cross products into the numerator and our 4 marginal totals into the denominator. Our final setup shows exactly how the raw data feeds into the statistical test, ready for computation. Now that we have set up the equation, let us work through the derivation of the final statistic to understand how the math proves significance. This chunk of the calculation focuses on the numerator, which captures the raw deviation of our data. Following the order of operations, we first simplify the difference of the cross products inside the parentheses, which gives us 300,000. Next, we square this difference. Mathematically, squaring ensures that our deviation is positive and heavily penalizes large differences, giving us 90 billion. Finally, we multiply this by our total sample size of 2,000, resulting in a massive 180 trillion. While this number looks intimidating, it is a necessary mathematical step to compute the final statistical weight of our genetic association. This sets us up perfectly to normalize the value in the next step. With our numerator calculated, we can now finalize the proof of statistical significance. We calculate the denominator by multiplying our four marginal totals together, which gives us 990 billion. This denominator acts as a normalizing factor, scaling down our massive numerator based on the sample sizes of our groups. Dividing the top by the bottom gives us a final chi-square value of approximately 181.8. In statistics, for this specific type of test, a value greater than 3.84 means there is less than a 5% probability that this association happened by chance. Since 181.8 is massively larger than 3.84, the math proves our intuition. We can confidently conclude that this SNP is genuinely associated with type 2 diabetes, and our initial odds ratio was not a random fluke. So, we have proven that our genetic link holds mathematically. This brings us to how scientists actually look at this data. What you are seeing here is called a Manhattan plot, the standard way to visualize genome-wide association studies. Let us walk through it. The horizontal axis represents all 22 human chromosomes, laid out end to end. Every single dot on this graph is a different SNP that was tested. The vertical axis shows the statistical significance, specifically the negative log of the p-value. The higher a dot is, the more statistically significant the association. Notice the horizontal dashed line. That is our threshold for significance, much like our 3.84 chi-square cutoff. Most dots are low-level noise, but look at chromosome 9. A massive tower of dots shoots up like a skyscraper, crossing the threshold. This visualizes exactly what we just calculated, a strong, undeniable genetic association. We have covered a lot of ground today, so let us review the key takeaways. First, genome-wide association studies are powerful tools that scan the entire human genome to find tiny variations, or SNPs, linked to complex diseases. Second, we learned that we can quantify this risk using the odds ratio, which tells us exactly how dangerous a mutation might be. Third, we saw that science requires proof, which is why we use the chi-square test to ensure our findings are statistically sound and not just random chance. Finally, we saw how Manhattan plots beautifully visualize these massive data sets. You now understand the foundational map that guides researchers toward better diagnostics and personalized treatments. Great job following along with both the biology and the math.